Hi, it's Miss Nicholas again. I'm going to talk to you a little bit today about spermatogenesis and the hormones that regulate the production of sperm cells. All right, so we'll begin with just an overview. You should have seen Dr. Ansardi's video by now about meiosis and comparing that to mitosis. Of course, hopefully you know that meiosis is going to be the process by which we create the sex cells, which in the male are the sperm cells and in the female the oocytes are the egg cell. So in this particular video we're going to focus on spermatogenesis, or the making of sperm cells. Genesis, of course, is always going to refer to the beginning of something. So the beginning of the sperm cells. So this is a sequence of events that's going to take place within the testes. And in the testes there are little tubes that are known as seminiferous tubules. So we produce sperm cells. I say we, I don't have seminiferous tubules obviously, but humans produce the sperm cells in the seminiferous tubules of the testes of the male. All right, so just a quick comparison and reminder that most of your body cells are going to be what are called diploid, are often referred to as 2N cells. Those are your somatic cells. Somatic refers to the body. So somatic cells have 46 chromosomes. One set of chromosomes you get from your mother's egg cell. The other set of chromosomes you get from your father's sperm cell. So you've got 23 pairs of chromosomes, homologous chromosomes. Homologous meaning that they are alike. Homo means same. Okay, so you get one copy of number 1 through 23 from mom, one copy of 1 through 23 from dad, and of course that gives you your 46 chromosomes. Gametes, however, are different in that they are what are referred to as N or 1N, and that is haploid, which means that they contain only one copy of those chromosomes. Now hopefully after watching Dr. Ansardi's video you recognize the importance of that and understand why. If at this point you do not understand the difference in haploid and diploid and why it's important that the sperm cells and the egg cells have 23 chromosomes, I would recommend that you go back and do a little review because it is important for you to understand those concepts. All right, so either you're up to par and you know what you need to know, or you've gone back and reviewed some information. So let's get moving on and talk a little more about the specifics. All right, so spermatogenesis, remember the making of the sperm cells. It's where we have cells that are located in the testes and they are going to give rise to the gametes, to the sperm cells. So this is a multi-step process that begins with mitosis. Now remember we talked about mitosis is not how we make our sex cells, our gametes, but the process does begin with mitosis. So we have cells that are known as spermatogonia, which are the stem cells that are located within the seminiferous tubules, and those stem cells are going to eventually make sperm cells. So we begin with mitosis, and remember when mitosis occurs, you are going to produce two genetically identical daughter cells. Okay, so here's our spermatogonia, and it's going to undergo mitosis and produce two genetically identical daughter cells. One of those cells, and we'll call that cell A, is going to remain a spermatogonia. The second cell, we'll call it cell B, is going to undergo meiosis and create the sperm cells. Now I want you to realize why that's important. If all of your spermatogonia began meiosis, then eventually the males would run out of spermatogonia because they would all be developing into sperm cells eventually. But by undergoing this mitotic division at the beginning of this process, it's constantly keeping a supply of these other cells. So later on, this particular cell is going to do the same thing. Undergo meiosis, create one cell that remains a stem cell, and create another cell that's going to undergo meiosis. So males have an unlimited supply throughout their reproductive history of cells from which they can create sperm cells. All right, so let's go back now to cell B here, our cell that's going to begin the meiosis, okay? And meiosis is going to produce the sperm cells. This cell, our sperm cell, is going to become a primary spermatocyte, okay? That little one with a circle means primary. That primary spermatocyte is going to undergo meiosis 1 and create 2 cells, which will both then undergo meiosis 2 to create 4 cells, and these are going to be your spermatids. Okay, spermatids, let's draw an arrow from here as well. Spermatids are not functional sperm cells. These are the last cells that are created by the division that is involved in meiosis, but they're not ready to do anything. They've got to undergo some specialization known as spermatogenesis. Now notice the difference in these two words. Okay, spermatogenesis is going to create your spermatids. Spermatogenesis is going to turn these spermatids into functional sperm cells. All right, so moving right along. All right, now here we have an image from your textbook showing you what we've been talking about. Ooh, boy, I stuttered over that, didn't I? Here we have the spermatogonium, the stem cell. So what you're looking at here is a cross-section through a seminiferous tubule. Okay, so your seminiferous tubule would continue on like this. All right, so here we have the sperm cell, or the spermatogonia, undergoes meiosis, creates that cell that's going to become a stem cell, retain the capability to undergo mitosis and create more and more. And then we have this cell, the B cell, or type B cell, that is going to eventually create those sperm cells. All right, so this type B cell here is going to change just a little bit to become a primary spermatocyte. So it enlarges a bit. It's going to replicate chromosomes, create a few new organelles, and then it's going to divide. And when it does so, it's undergoing meiosis 1. Now remember, back up here we have mitosis, okay? So meiosis 1 creates your 2 secondary spermatocytes, okay? So those are your secondaries. Those are your primaries. Here we have secondaries, okay? All right, now your 2 secondary spermatocytes are going to undergo meiosis 2 to create your early spermatids. Now notice from one primary spermatocyte, we ended up with 4 spermatids. So those 4 spermatids will then create 4 sperm cells. So from every cell that begins meiosis in the male, you end up with 4 sperm cells. Now the way to remember this in their divisions, primary means 1, secondary means 2. So your primary spermatocytes undergo meiosis 1, your secondary spermatocytes undergo meiosis 2 to create your spermatids. Now your spermatids are going to change. You see here you have early spermatids, just little round circular cells, and then you have late spermatids, which begin to look a bit more like sperm cells. So you see there you've got some structure to it as well as tails. Now this particular image labels these as spermatozoa. Spermatozoa are your functional sperm cells, but only a few, and I mean very, very few, are actually going to become spermatogonia, or spermatozoa, I'm sorry, in the seminiferous tubules. Okay, now remember it's that, excuse me, my nose is itching. It's that specialization, that spermatogenesis, that makes functional sperm cells. That doesn't occur in great quantities within the seminiferous tubule. It occurs in the epididymis, which is located on the posterior and somewhat anterior aspect of the testes. So here, of course, you're going to have a lumen inside your seminiferous tubule. The lumen is the opening in the middle of a tube. So you're going to have spermatogenesis occurring all out in here, and you're going to end up with these little spermatids with their tails in the lumen. Now, most of your spermatids are going to leave the lumen and travel out to the epididymis. And I'm just going to abbreviate there. The epididymis is where spermatogenesis, or the maturation of the sperm cells, occur. Okay. Now, to give you a different look at the same thing we've just looked at, let's look at let's look at an image taken with a photomicrograph. So it's a microscope and an image taken from that of a cross-section through a seminiferous tubule. All right, so if you see the G's are spermatogonia, your I's, and it took me a while to find it, over here's your I, and then the Z for spermatozoa, okay? So let's look and see what we've got here. Your spermatogonia, your stem cells, are going to be these small little cells around the edge. So these are on the outer edge of the seminiferous tubule, okay? They're going to undergo that mitosis and specialization and produce some primary spermatocytes. So you see these are much larger cells. These spermatocytes undergo meiosis I to create smaller secondary spermatocytes. And you've got several layers of those in here. Those are your secondaries. And then your secondaries are going to undergo meiosis II and create even smaller little spermatids. Now you see some of these spermatids are going to have their little tails attached already and are going to start the maturation process. Now I also want to point out while we're looking at this image, these patches of cells that are outside of the seminiferous tubules. Okay, outside of the seminiferous tubules, you have interstitial cells. Your interstitial cells are going to be important when we begin talking about those hormones that regulate this process. So remember this image when we start talking about those cells. And let's move on. All right, so we've gone through an overview of how the sperm cells are produced. Excuse me, let me refer to my notes, make sure that I'm telling you everything I want to tell you. And I am going to change colors of pen to blue so it will show up a little better on this background. Okay, so let's talk about the hormones that regulate this process. Of course, the reproductive system is reliant on hormones to drive the production of the gametes. So from your study of the endocrine system, you should be familiar with the hypothalamus and the pituitary gland. We're referring primarily to the anterior pituitary gland. So let's get familiar with what we're seeing here. Here we have the hypothalamus, okay, the infundibulum connecting the hypothalamus to the anterior pituitary. All right, down here we have a seminiferous tubule. Now remember, we just looked at the seminiferous tubules, and then this little patch of cells here are your interstitial cells outside the seminiferous tubule. Now hopefully you could tell me what the hypothalamus and the anterior pituitary, what roles they play in this process, or at least have some familiarity, because you should have heard about gonadotropin releasing hormone from the hypothalamus and FSH and LH from the anterior pituitary. So you've got gonadotropin releasing hormone is released from the hypothalamus, travels to the anterior pituitary, and tells the anterior pituitary to release follicle stimulating hormone and luteinizing hormone, which then travel to the gonads. In this particular case, we're talking about the testes. Okay, so hopefully that was a little bit of review for you. So let's follow along and see how this happens. In the male, every 60 to 90 minutes, you have a surge of gonadotropin releasing hormone. Okay? That surge of gonadotropin releasing hormone is going to cause several events to occur. Hopefully after this you can see why guys are always horny. See guys, I'm giving you a reason. I'm giving you something to argue. I can't help it, honey. It's because my gonadotropin releasing hormone is constantly causing surges of testosterone. Yeah, you can pay me for that later. All right, so let's get back to where we're at. Your hypothalamus releases gonadotropin releasing hormone, step 1, okay, to the anterior pituitary. The anterior pituitary, then step 2, and we're going to follow this along sequentially here, is going to release luteinizing hormone. Your luteinizing hormone targets the interstitial cells and tells those interstitial cells to secrete testosterone. All right, now let's hold off right there for just a moment and go back up here to number 3. At the same time we're releasing the luteinizing hormone, we're releasing follicle stimulating hormone. The follicle stimulating hormone binds receptors on the outside of these support cells. Okay, so you see this big gray mass here. Okay, this particular gray mass on this image is one of the support cells known as a sustentacular cell. And let me write that out for you, sustentacular, sustentacular or Sertoli, named after someone that discovered them, cells. Okay, these cells surround your spermatogenic cells. So these cells are the ones we've been seeing that are eventually going to create your sperm cells. Your sustentacular cells are support cells that play a very important role, as we'll discuss here as well as moving on. So let's go back now to our number 3. The follicle stimulating hormone is released and is going to bind a receptor here on the sustentacular cell. It causes that sustentacular cell to make a protein known as androgen binding protein. Now remember, testosterone is an androgen. Okay, so your androgen binding protein is going to bind that testosterone and create a complex. Okay, so that complex made up of your androgen binding protein and your testosterone then travels back to the sustentacular cell and stimulates those spermatogonic cells to begin making sperm cells. Okay, so far so good? All right, the other thing that's going on is when your FSH binds that receptor on your Sertoli cells or your sustentacular cells, they also start to secrete a hormone known as inhibin. Now surely you could figure out what inhibin does. Inhibin inhibits the release of more hormones. So you slow down the production of sperm cells, because if you're producing sufficient amounts, because you just released FSH and LH, you don't need to be making more until the levels drop again. All right, so we're making that inhibin, which has got a negative feedback reaction or controlling mechanism over the hypothalamus and the pituitary gland. Now let's look at what else is going on with that testosterone. Remember, we had some of the testosterone that binds to the androgen binding protein, which then stimulates spermatogenesis to occur. Some of that testosterone travels out to other targets in the body and is responsible for maintaining the secondary sexual characteristics. Your secondary sexual characteristics are the things that you look at that tells you whether that body is a male or that body is a female. So if you think about what the male secondary sexual characteristics are in a male, you notice the increased muscle mass, you notice hair on the face and the underarms and the chest, you notice that the muscle mass is different. I said that already though, didn't I? The testosterone is responsible for, during puberty, of course, the maturation of the testes and the penis so that they grow to the mature size. It's responsible for the libido. That's why you're horny all the time. All right. So you know about testosterone and what it's responsible for. So now we've seen two things that this testosterone is doing. One, it's bonding with the protein and stimulating spermatogenesis. Two, it's maintaining the secondary sexual characteristics. And three, that testosterone also helps the inhibin with the negative feedback. All right, take a few minutes and go through that. This is a really, really good little illustration of the way that these hormones play a role in the development of sperm cells. All right, let's continue talking now about those sustentacular cells. Remember, they're also known as the Sertoli cells. So they extend from the wall of the seminiferous tubule to the lumen, or the opening of the seminiferous tubule. They provide nutrients and signals to those cells that are dividing. And they do so, of course, the nutrients are going to be because the cells take those nutrients in, metabolize the nutrients and get them out to the developing spermatogenic cells. And the signals that we were talking about, of course, are going to be the inhibin. And let's back up to that just a moment. So you see that you've got the inhibin that's going to be released, and you've got the androgen binding protein that is going to bind with the testosterone and then come back down and stimulate spermatogenesis. Dispose of excess cytoplasm that's sloughed off during spermiogenesis. Now remember, that's on a limited basis, okay, because most of spermiogenesis is going to occur in the epididymis. And we'll talk a little bit more about that on a slide that's coming up. And also, your sustentacular cells are going to secrete a bit of testicular fluid into that lumen. Now remember, here's your seminiferous tubule. You've got your sustentacular cells in here, and you've got your spermatogenic cells here. Your lumen is the opening. So it's going to secrete some fluid into the lumen, into the opening, so that that sperm is easily transported out of the seminiferous tubule and onto the epididymis for spermiogenesis. But another really important thing that they do is to provide a tight junction that forms a blood testis barrier. Now let me take you back to an image that we saw just a few minutes ago. Let's go back here. Whoop, too far. Let's go back to this one, okay? So here, and let's erase all this. Bear with me. There we go. All right, so here you have the nucleus of a sustentacular cell, and here you have a nucleus of another sustentacular cell. Okay, so here are your tight junctions between those cells. All right, a tight junction, remember, is going to hold cells tightly together and prevent things from passing from one cell to the other. So this allows this whole compartment to be divided into a basal compartment, okay, everything above your tight junction, and an adluminal, near the lumen, an adluminal compartment, which is everything below your tight junction. Now this is important. When you think about providing a barrier, surely you've heard of the blood-brain barrier before, where you have tight junctions between cells that prevent things from leaving the bloodstream and getting into the brain. Of course, some small molecules get across, as well as some gases and nutrients, but the blood-brain barrier is there to only allow certain things to cross. Well, the same thing is important here in the testes. Now, hopefully you've studied the immune system and you know a little bit about what the immune system does. During development, the thymus is important for educating your immune system, and it does so by sort of taking an inventory of all of your antigens, and you know that antigens are proteins that are found on your cells. Well, you have these proteins, these antigens, on all of your body cells, including your spermatogonium and your spermatocytes and your spermatozoa, okay? But when you're undergoing this thymic education or doing this inventory of your antigens, this is going on during fetal development as well as during young childhood. Well, these sperm cells are not there during young childhood. Remember, this whole process of meiosis begins at puberty. Well, you've already programmed your immune system so that the immune system knows what to look for. Now, this tight junction prevents the immune system from recognizing these cells that are just being formed at puberty. Okay, now these cells, your spermatogonia that are up here, your immune system recognizes those because they are present during development. But your spermatocytes only become in the body, or only are made in the body after meiosis occurs, and that's when your hormone levels rise enough to trigger this to begin. Okay, so it's important that these cells are kept away from the immune system, okay? Because your immune system would see those antigens on those spermatocytes, never seen them before, and think that those are antigens on a particular pathogen. You know, and the immune system, when the immune system finds a pathogen, they try to destroy the cells that contain those by creating antibodies and launching an immune response. So if the immune system sees these, they don't recognize it, they mount an immune response, and these cells all die. Dead sperm cells can't fertilize eggs. Okay, so the blood testis barrier that is provided by those tight junctions between your sustentacular cells hides those developing sperm cells from the immune system. Pretty cool stuff, huh? All right, let's get back to where we were and talk I think we covered everything on that. Absolutely, I did. Okay, let's finish up talking just a little bit about spermiogenesis. Now, spermiogenesis, remember, occurs in the epididymis. Okay, the epididymis, if you look at an individual testis, okay, here would be the posterior surface. The epididymis, let's change colors there. This would be, I said change colors, go to red. This would be the epididymis, okay? So located a bit superior and posterior to the testis. And then, of course, it would continue on to be the vas deferens, that would become the urethra, and then the urethra, of course, would exit the penis, and that's how the sperm cells get out. So let's talk about this whole process of spermiogenesis. Now, we start off with these spermatids, remember? Okay, so this is an early spermatid. And we know that because it's still round, it really hasn't done anything. But let's think about what the job of a sperm cell is. The only thing a sperm cell needs to do is swim, find an egg, and drop off that DNA. And of course, you know that the DNA is found within the nucleus. So here, this little purple structure is the nucleus of the cell. So all the DNA is found within there. But those little early spermatids are not going to get anywhere. Look at them. They've got to first develop a tail. So the tail is a flagella. Okay? Or the tails are flagella. One is a flagellum. Okay, so that's singular. Flagella is plural. So we've got to, one, get the DNA to the egg cell. We've got to swim. And if all that's going to happen, you're going to have some energy. What is it in a cell that provides energy? Of course, it's your old friend ATP, right? Well, if you're going to make ATP, you've got to have somewhere to make it, and hopefully you know that that is in the mitochondria. So the mitochondria are responsible for making the ATP. So if you look at this particular image again, you see all these little mitochondria in there. All right, so we need to get all those mitochondria into one particular area and wrap them around that developing flagellum. All right, now all we need, remember, is the DNA. So we've got the DNA in the nucleus, and we've got the mitochondria so that we've got energy. We've got the flagellum to power. Okay, so that's going to propel, and it's going to twist, sort of. And as it does so, it's going to propel the sperm cell along the vaginal canal up into the uterus and out to the fallopian tubes, hopefully to find an egg. But now let's think about this. When we're making these early spermatids, we've got all this bulky cytoplasm. We don't need all of that. So we've got to get rid of it. We've got to shed that cytoplasm. We've got to get all those mitochondria around that flagellum, and we've got to develop that long flagellum and get ready to go swimming. So why do we need to get rid of all this bulky cytoplasm? Well, let's think about it like this. Suppose you're going to go swimming, and it's in the middle of winter, or whatever time of year, doesn't matter, but you're dressed in winter clothing. So you've got on your heavy boots, and you've got on a pair of jeans. Underneath those jeans, you've got a pair of long johns. You've got on a long john shirt, and then you've got a t-shirt, and then you've got a hoodie. And on top of that, you put your winter coat on, and it's got a big old hood, and you got big old bulky gloves on. And I tell you, go jump in the river and swim across from one side to the other. Well, you go on down there and you jump in, and what's going to happen? Your butt's going to sink. You're not going to be able to swim. Okay, you've got too much stuff on you. You're too bulky to swim. Well, that's sort of what all this mess is on these cells. All that excess cytoplasm is going to prevent the sperm cells from being able to swim. So if I told you now, go down to the river, take off all your clothes, jump in and swim across, you're going to be much more capable of swimming. Okay, might draw a crowd, but at least you can swim, right? So that's what's going on here with all this excess cytoplasm. It's being shed to where at the end you have a functional sperm cell with a flagellum, a mid-piece here with all the mitochondria, and you have the head ready to swim. All right, one last slide to go over, and let's talk a little bit more about those particular areas. All right, so we've got the flagellum, okay, that's our tail. All right, so that's the motor that powers the swimming. We've got the midpiece. I wanted to change colors. Let's go with black. Okay, so you've got the midpiece here with all your mitochondria. Okay, and then we've got the head of the sperm with our DNA. But there's one more thing that we've got to have. Now, egg cells are very well protected. All right, so those egg cells are going to be ovulated from the ovary, and they're out there in the fallopian tube, but they've got a tough outer covering around them known as the zona pellucida. That zona pellucida prevents things from getting in and damaging that egg as it's moving along the fallopian tube. So all these sperm cells that are being ejaculated, and there are millions of sperm cells in each ejaculation, they've got to have some way to get in. I mean, they can't just walk up and ring the doorbell and say, "Hey, egg, it's me, let me in." So we've got to carry something with us in order to get in. So the sperm cells have a little toolbox known as an acrosome. That is a cap-like structure over the head. There it is, acrosome. Okay? So the acrosome contains enzymes that are going to digest away. We'll draw some green little enzymes in there. Okay? Those little enzymes in there are going to digest away that tough outer covering on the egg. So you have millions of sperm cells that make it to the egg cell. They all start dumping out the enzyme. The enzyme, I'll go ahead and give you the name, is hyaluronidase. Hyaluronidase. How did you know that was an enzyme? ASE, right? So hyaluronidase is the enzyme that all these sperm cells dump out on that zona pellucida and digest it away. The one particular sperm cell that happens to be at the spot that gets digested away first gets in. But only the head of the sperm cell gets in. We don't need the mitochondria. We don't need the flagellum. All we need is that DNA to get in, right? So the DNA in the head of the sperm cell gets in. Immediately, zonal inhibition proteins, often called ZIPs, are going to cause that zona pellucida to reharden. When that zona pellucida rehardens, it prevents more than one sperm cell from getting in. Because remember, we've got 23 chromosomes in the sperm. We've got 23 in the egg. That's going to give us our 46 chromosomes. We don't need another sperm cell getting in, bringing another 23 chromosomes, right? It'd be bad news. All right, so I think that's everything I wanted to tell you. Hopefully you've got a better idea now of spermatogenesis and how sperm cells form. I'm sure your instructors will talk to you a little bit more about it, and I would strongly recommend that after watching this, take your textbook and go to the particular section in your textbook about spermatogenesis and read over that and make sure that you understand the role of the hormones in this process as well. I'll see you next time.